So good afternoon, everybody, and thank you for joining this webinar. I am Sara Pittonet, the coordinator of the Blue Cloud Initiative, and I'm happy to welcome you to this series of webinars dedicated to the Blue Cloud demonstrators. And today we will discover the Plankton Genomics demonstrator. So, Blue Cloud is the flagship initiative of the Horizon 2020 Future of the Oceans and Seas program of the European Commission and aims to promote the sharing of data, processes and research findings in the marine domain by delivering a collaborative web-based environment that enables open science, underpinned by simplified access to an unprecedented wealth of marine data resources and interoperable added value services and products. The project started in October 2019 and has a budget of 5.9 million euro. It includes 20 partners and 13 blue federated infrastructures. So one of the components of this technical framework are the Blue Cloud virtual research environments that provide computing platforms and analytical services to facilitate the collaboration between researchers. In particular, each Blue Cloud VRE includes services that facilitate collaboration, services that support analytical tasks in a distributed computed infrastructures, and services that enable the co-creation of new entire virtual laboratories. 5 Blue Cloud demonstrators are developed, making use of existing services and driving the further development of additional Blue Cloud services. Today, you'll meet the Plankton Genomics demonstrators. That is one of the 5 demonstrators that the Blue Cloud is developing, addressing specific challenges tackled by researchers in biodiversity, genomics, environment, fisheries, and aquaculture domains. So the demonstrator Plankton Genomics aims to showcase a deep assessment of plankton distributions by mining data across biomolecular, imaging, and environmental domains. It consists of 2 Jupyter notebooks that will be presented in the webinar today. The demonstrators is led by the European Bioinformatics Institute and created by the Faculty of Sciences at the Sorbonne University with contribution from the Flanders Marine Institute. So the Blue Cloud platform features several services that can be used for undertaking world-class science via the European Open Science Cloud framework. So for those of you who are familiar with the concept of the EOSC, the Blue Cloud project addresses the EOSC challenges in different ways. So first of all, the platform, the cyber platform that Blue Cloud has developed provides fair access to multidisciplinary data sources, and it constitutes a pilot thematic EOSC model that can be replicated for other thematic clouds. And it also introduces a methodology for researchers that want to interact with infrastructure developers. But Blue Cloud also contributes to address several societal challenges defined by different policies at different international level. And it tackled the issues of interoperability across different marine community services and in general across different thematic functionalities. So providing easy access to services and tools, introducing the demonstrators as practical sample of implementation of the release, and offering a catalog to access the services that will be integrated with the EOSC portal catalog. Last but not least, the Blue Cloud is bringing the EOSC in the blue economy through its policy-oriented roadmap that will be launched very soon in June. Blue Cloud is synergizing from the very beginning with other initiatives and actors in the marine domain, establishing relations to different purposes, so to boost the usage and exploitation of its theories and services, to connect data across different providers and across different services and sectors, and also to gather interest in the Blue Cloud roadmap and the sustainability of its services. And for that reason, in particular, is also liaising with the initiatives outside Europe at the international level. As I mentioned before, we are launching the Blue Cloud roadmap in June, and there will be an open consultation where we will ask contribution and feedback from all the stakeholders interested in supporting and providing a vision for the sustainability of the Blue Cloud services. So today, as I mentioned, you'll have a deep overview of the demonstrators of Plankton Genomics. I'll now leave the floor to Stéphane Pesant, who will introduce the demonstrators. And then he will be followed by Pavla Debeljak and Jean-Olivier Irisson from the Sorbonne Université, who will provide an overview of the notebooks. With that, we wish you a very interesting webinar. We invite you to ask any questions you might have using the question and answer tab in your Zoom window. And feel free to get in touch with us through all our channels.
Thank you, Sara. Let me share my screen. So hello, everyone. We thought that we should start by explaining the inspiration behind the demonstrator. So it all started with the Tara Oceans expedition. It's an expedition organized by 20+ organizations, and it happened on board the sailing research vessel Tara. And on that vessel in 2009 to 2013, we did a global expedition. We did a total of 210 stations covering most oceans, going across a wide variety of environmental conditions. And so during the expedition, we used a number of standard oceanographic instrumentation, including, let me use this, including the Rosette sampler that you see here, which was equipped with a number of physical, chemical, optical sensors, and it was collecting water as well to prepare some samples. And we also used nets, a number of different types of nets to collect larger organisms. And on board, we used several protocols in different labs to prepare samples, mainly doing filtrations, concentrating water, collecting organisms into a large number of samples. And so the objective was to study plankton. Why plankton? Because they represent roughly 60% of the biomass in the global ocean. They feed most life form in the ocean. They drive the carbon pump, and they are largely unknown. More than half of the diversity of plankton is yet to be discovered. And in Tara Oceans, we looked at the full size spectrum of plankton, going from viruses to bacteria to phytoplankton, zooplankton. So we tried to cover the full ecosystem of plankton. The protocols that we used targeted two main groups of analyses. The first one being high throughput sequencing. So sequencing, preparing samples to sequence single organisms, but also looking at community level, so sequencing DNA to look at community diversity, sequencing RNA to look at community or functions that are present in the system and mediated by plankton. And the second group, we had lots of samples that were then analyzed in the labs using high throughput imaging techniques, using a number of instruments in order to cover organisms from the full size spectrum. Then the data from all these analyses are archived in long-term and open access repositories, mainly, but not only, but mainly at ENA, the European Nucleotides Archive for the sequences, and the images at the Bioimage Archive, plus local archives as well. And then the environmental data is, they are archived in a number of repositories, including Pangea, CDataNet, and ultimately EMONNet for the more chemical and some biological data as well. And we also used climatologies and satellite data from Copernicus. So all these data are in open access, and they have been used intensively by people in the consortium of Tara Oceans, but more and more they are being used by people outside the consortium. And they're used to generate a number of analyses, pipelines set up to look at the biogeography of assemblages, looking at how they correlate with the environment, looking how organisms interact with each other, how they are associated with each other. And all these pipelines, let's say, were developed by individual scientists or groups of scientists working with their own resources locally. And now the dream of the Blue Cloud is to be able to deploy these analyses pipelines in the cloud, so that anyone can go in the cloud and either redo the analyses that were published or use the pipelines with their own data, or even take the pipeline, modify it, tweak it so that it can be fit for their purpose, and then put it back in the cloud. That's the dream. And so we came up with this Plankton Genomics demonstrator developed by a number of people from EMBL-EBI, from Sorbonne Université, and from VLIS. And today, Pavla will tell us about the state of this demonstrator, what can be done at the moment, and Jean-Olivier will tell us about the next steps. And with that, I will leave the floor to Pavla. Let me unshare.
Thank you. Sorry. Yes, I'm going to present the state of the art of the Plankton Genomics demonstrator. So this is, as Stéphane said, a collaborative effort between Sorbonne Université, the EMBL, the VLIS, and the CEA. I'm just going to briefly introduce what kind of data we have integrated into it. So Plankton Genomics describes omics, which in general is the study of whole genomes of organisms. So in this case, this would be Emiliana Huxley, and we want to study the total DNA of Emiliana Huxley as an organism. However, in the Plankton Genomics demonstrator, we actually implement meta-omics datasets. So it's the study of genetic material recovered directly from environmental samples, which here would represent a quite diverse community, which we can extract DNA and RNA from. So these meta-genomic and meta-transcriptomic datasets, they represent the total environmental DNA from organisms that have been sampled during Tara Oceans, and the total environmental messenger RNA. So it's the part of the DNA that is transcribed into messenger RNA, which would later be translated into protein. And through this sequencing that has been done, we obtain metagenomic and metatranscriptomic datasets. These data sets are then annotated through public databases. So what has been done on these Tara Ocean data sets is the annotation of protein families. So protein families which combines multiple sequence alignments and hidden Markov models. And this is for the functional annotation. So what this gives us is the known functional aspects of the metagenomic and the metatranscriptomic data sets. And the database that has been used for the taxonomic identification is the Marine Microbial Eukaryote Transcriptome Sequencing Project, which was enlarged with other eukaryotic plankton group sequences. And a weighted least common ancestor algorithm was used to annotate taxonomy. So we basically have two data sets of known function and known taxonomy. As Stéphane has mentioned before, so when we look at just the Tara Oceans data, we look at the data set of the marine eukaryote. This is the map of the 68 different sampling locations in all the different oceanic provinces for the surface waters and the deep chlorophyll maximum. We can only identify about 50% of these data sets. So it means that 50% is the so-called unknown or not annotated or hypothetically not annotated part. And it has been common practice to not look at this actually in most of the omics data sets because it is reoccurring in environmental data. So in the Plankton Genomics demonstrator, we want to implement this and want to use this data set. And so it is divided into two notebooks. So first of all, the discovery of this yet unknown biodiversity from these unassigned genetic signals in comparison with the known biodiversity. And what is quite interesting, I think, is that we combine this within the environmental context, so with the rich environmental metadata that Tara Oceans provides. And the second part of the notebook is the habitat modeling and biogeography of these not yet described plankton diversity. So I will now present the sort of data sets that are used as input and the sort of data sets that can be obtained through these pipelines. So the Plankton Genomics demonstrator as it is now is based on this marine atlas of Tara Ocean unigenes, which is a huge collection of 116 million genes. What we can implement at the moment in the demonstrator is 10% of the gene catalog. So this is implemented in the virtual research environment of the Blue Cloud. And to this, we have added the associated environmental data. The aim is to provide really a framework for the exploration and easy access of omics data sets. As Stéphane has mentioned before, Tara Ocean used different size fractions. So I will go through 4 examples in these different planktonic size fractions to highlight the data exploration that can be done. I will also look at the epipelagic layer, so the first, the upper layer of the ocean that has been sampled during Tara Ocean, so the surface, the deep chlorophyll maximum, and the mixed layer. There are multiple others, but this is the part that has been implemented at the moment in the notebook. This is how the workflow actually looks. So the input for the demonstrator notebook 1 are 4 data files. It's the known taxa, the known function, annotated function, and the gene catalogs, the mapped gene catalogs of the metagenomic and the metatranscriptomic data. So again, the metagenomics is the genetic potential, the metatranscriptomics is the gene expression. And the notebook 1 then basically includes the Jupyter notebook for known and unknown exploration. And this can then be passed to our markdown files, which are implemented in the Jupyter notebooks for statistical analysis and visualization. And all of these data sets that I will show are published in the Blue Cloud catalog and can be downloaded and retrieved with the associated data. So the first size fraction I will look at is the picoplankton, so 0.8 to 5 micrometers size fraction. A representative of this group of the known annotated is, for instance, Ostrococcus tauri. It's not very abundant in the ocean, but it is known as the smallest eukaryotic entity on Earth. And in this size fraction, for instance, we can extract everything that is picoplankton. And we can then look at the known and the unknown fractions and calculate the ratio for different parts of the ocean, for different stations, or for the different data sets. In this example, we see for the picoplankton, we have more functional unknowns. So we divide the known by the unknown for the same data set. And when we look then in the different depth layers, we see the whole, all of the surface depth layers show a high amount of functional unknowns compared to taxonomic unknowns. So this is, again, one result where you can further on build up theories or hypotheses. What we can do then is look into the horizontal coverage. So to briefly explain, when we look at a chromosome on a DNA fragment of one organism, we can map shorter elements to this sequence and look at how often it was actually sequenced or how often it was actually transcribed. And in the case of the MATU catalog, we have one big catalog of genes that has been assembled out of these short sequences that have been obtained. This we call the contigs, so the consensus DNA sequences. And when we talk about environmental omics, we can map them back to this gene catalog. So we map back the short sequences from the metagenomic data or the metatranscriptomic data to the big gene catalog that has been provided of all eukaryotic plankton that was found during Tara Ocean. And what we get then is the horizontal coverage. So the idea is to have an abundance value for one gene or one transcript. And in the case of the pico size fraction, when we look at the horizontal coverage in the y-axis for the knowns, so these are the annotated taxa, so the species that we know. We have the DCM and the surface layer, and on the x-axis you have the Tara stations, and the Tara stations from 0 to over 150. And we see that, well, in DCM we have some that have a higher horizontal coverage, but it's nothing compared to when we look at the unknowns. So this again shows us that these unknowns, so this part of the diversity and the transcriptomic diversity as well in the functions, seems to be extremely abundant compared to those that we know, meaning that what we cannot identify has some functional importance or could have a potentially big role in our oceans. So these plots and all of these codes are provided in the demonstrator notebook 1. And so that's the first part, is basically these datasets can be retrieved from the catalog. They are the known and the unknown datasets by size fraction and by depth. For the next size fraction, so we look at the nanoplankton from 5 to 20 micrometers. An example would be, as I said before, Emiliana Huxley, so this quite beautiful coccolithophore, which we know has very diverse morphological structures, and is a bloom former, so it's known for these big blooms in the English Channel. What I'd like to showcase here is, until now I've been talking about the metagenomes, so we can add the metatranscriptomic data. Here is a plot that actually shows the metatranscriptomic versus the metagenomic data. So it's somehow like in when you would have a culture and you would look at the DNA versus RNA. And if we compare this to the known and the unknown, we kind of see a decoupling between these two data sets, which seems to be higher in the nanoplankton fraction as well for the unknowns. So again, this is another point where this can be done for all size fractions, all depth layers, all different data sets to identify what is of interest for a specific plankton group. And we can then add, for instance, in the DARA data, the environmental data. So can easily plot any kind of horizontal coverage with the metatranscriptomics or metagenomics against any of the environmental parameters. So whether nitrate or phosphate, temperature or silicate, whatever is of interest for the specific group. And in this case, I'm showing you the metatranscriptomes against the nitrate concentration. So you see that several of these sequences seem to have very high horizontal coverage with quite low nitrate level. And the notebook can pinpoint exactly to which station, and this station can then be plotted. And again, it is more of a hypothesis-driven tool. So from here on, you can postulate further experiments. And this as well is implemented for all the size classes, and all of these data sets with the corresponding environmental data can be downloaded from the Blue Cloud catalog. I will now combine the last two size classes. So there's the microplankton, which is from 20 to 180 micrometers. An example would be the Tintinida with these beautiful morphological forms that actually have, through sequencing, been identified as an extremely diverse group, as they have only been defined by their shells before. And a very large fraction of the mesoplankton are the copepods in the Tara Ocean. So in the larger, bigger size fraction, directly consuming phytoplankton. So in this last example, we can add the information of the knowns and the unknowns by sampling site. We can then calculate these ratios, and we can plot the ratios corresponding to different size classes. Or we can also, as in the final example, you can have interactive maps where basically here I plotted the ratio between meso or microplankton in the different surface layers, which are shown with the different color code. And to each station you can point and you will find all the environmental data, station number, gene length, metatranscriptomic and metagenomic information. And this is also implemented in the Python notebook of the Blue Cloud demonstrator. Yes, so that's the last part. These interactive maps combining these environmental data of the Tara Ocean, which again are all to be downloaded, and the codes themselves or parts of the code can be downloaded as well. But until now, I've been mentioning the parts or the analysis parts that have been classically done, so correlation analysis with environmental parameters, which is when we talk about unknowns as far as we can usually go. There are two other approaches to focus on more of the functional aspect of these unknown sequences. One of them is the function unknown families, so trying to find the robust domains of these protein families that are really bona fide new functions through a homology search, which was published in a paper in 2018. And another one would be the sequence similarity networks. So I will just briefly explain these. So the notebook includes these eukaryotic functional families of interest from this paper in HMM format, so they can be downloaded, and HMM search, so bioinformatic tool, can be used to identify them with their individual FASTA sequence. So this is now not anymore focused only on the Tara, but really just an analysis in one own FASTA sequence format. Here, for instance, I've tried this with the Tara Ocean data as well. So for 2 different stations, and this can be then compared in different size fractions for the gene length and the horizontal coverages. The notebook also contains the code for the possibility to create sequence similarity networks. So sequence similarity networks is really the next tool to be used to compare different data sets, because the Tara Ocean data is great as a resource, and it has been a real pleasure to implement it, as it contains a lot of information and a lot of depth to the data is given with the environmental data. However, in the future, it would be interesting to combine multiple other data sets from other sources or from other cruises or even from other sequencing technologies. And the way to do this is through sequence similarity networks, as they can combine heterogeneous data. And so these two are also given as codes in the Plankton demonstrator and can be implemented in RIgraph or even in Python. So this is how the entry to the virtual lab looks like. This is the link to it. And so it provides these tools to analyze the known and the unknown sequences out of the MATU, so the Marine Atlas of Tara Ocean Unigenes, and the corresponding environmental data. As I said before, all the codes and data sets used are available, and the generated data can be retrieved from the Blue Cloud catalog, which can be of interest if you're focusing on a very specific group of plankton, whether it's known or not known. And I would like to thank for your attention. And these are all the people implemented in the generation of the demonstrator. And I can pass on to Jean-Olivier.
Thank you. So, okay. So thank you everyone for being here, and thank you everyone for the presentation so far. So I will give a really quick presentation of the next step of that demonstrator, which is the extrapolation of what Pavla just presented to unsampled parts of the ocean. Because Tara Oceans, as Stéphane presented, is a global cruise. Okay, it sampled almost all ocean basins, but it has local stations. Okay, so point observations in certain places in the ocean. And what we would like is, from the analysis that was done at this point, to try to guess what happens in places that are not sampled, like the middle of the Atlantic, for example. So what we start from is something like this. Okay, we have a map with some points where we have some information, and let's say that the size of those points represents the amount of a specific unknown that is of interest to us, okay, because it's particularly abundant in some places or in some environmental conditions. What we would like to have is a map like the one below, so something complete, where the color codes for the abundance or the proportion of that particular unknown sequence. Okay, so this complete map can help us in several ways. It can help us trying to guess what that unknown is by inferring, okay, yes, it's present in those types of conditions, it's present in those types of oceanographic structure. It can also help us guide future cruises, saying, okay, this particular unknown, it's really abundant in some condition. We would really want to sample it more particularly. Where should we go and when should we go to sample it? Okay, so the question really is, how do we go from that local information to a more global map like this? And one way to go from this to this is by assuming that plankton or some functions of plankton really depend on the environment. Okay, and that's one characteristic that's quite often very true for planktonic organisms. So because it depends on the environment, and because we have complete maps of environmental conditions from satellites, from global models with data assimilation, etc., what we can use is use this environmental information to make a link between what we know and what we would like to know. So, for example, for a given station, we can go in the environmental map and retrieve so in that case, the temperature value for that station, and plot the temperature, the observed abundance as a function of the temperature. And we would do this for every single station. So in Tara Oceans, we have about 150 of those. Okay, and then from this, we can draw that curve, or we can use statistical tools to draw that curve that represents the potential habitats of that species. Okay, when the temperature is low, then we don't have a lot of it. When the temperature is high, then we have a lot of it. So I'd said species, but in our case, really, it's going to be some genetic sequence that we can identify as being an entity, but that we don't know what it is. And that sequence can be representative of a taxon, but it can also be representative of a function. Okay. So once we have derived that curve, what we can then do is say, okay, let's go at a point where I haven't been sampling. And then from that curve reads the predicted abundance of that particular entity. And we can do that for every single point in the ocean, and then we get that complete map. Okay. So in practice, of course, we don't use just one predictor like the temperature here. We would use multiple predictors and build here a multivariate model that links the abundance, the environmental data to predict the potential abundance everywhere in the ocean. Okay, so that process is quite classical now. It's called habitat modeling or niche modeling when it applies to species. Again, we won't be dealing with species here. And of course the goal is to do this in the blue cloud, but that is something that has been done quite a lot, so we're confident that we're going to manage to do it. Some possible innovation that we would like to bring, and it will be the job of the person presented here, Alexandre Chekelot, who will start a postdoc very soon with us on this topic. So there's two innovations. The first one is that, as Pavla presented, what the analysis gives us is not just a single entity, but it's the relative abundance of many different entities. And there is information to gather from this. For example, in this story example, we have 4 stations and we have 3 entities of different colors. And what you see is that when the yellow entity is high in abundance, then the blue entity is low in abundance. And on the contrary, when the blue is high, then the yellow is low. So it means that instead of predicting them one by one and potentially predicting things that do not occur in nature, like the two, the blue and the yellow high at the same time, what we can do is try to predict the whole community or a subset of it together, and to, in that case, exploit the additional information that we have from the observed relative abundances. Okay, so that's one thing that we're going to try to extrapolate more efficiently from 150 stations, which is of course a huge effort in terms of sampling, but is actually not very much in terms of machine learning. The second aspect is that from Tara Oceans, we have local conditions, really precise conditions together with the station. But actually what you get when you sample in a point in the ocean is a really partial view of the context. So what you have here is a satellite image that shows a really nice vortex with a bloom. And what you see here is that if you sample right at the middle, or if you sample at the edge, you would be in quite different conditions. So what we would like to do is to capture that kind of mesoscale environment, to say, okay, that point where we sampled plankton, it was actually within an eddy, it was outside of an eddy, it was near front, okay? And one tool that has recently been developed, that has been recently used for this, is to use deep learning. So deep learning is a new kind of machine learning that works from images, and that is really good at extracting structure from an image. Okay, and so in that case, so deep learning is really often used to classify images. Okay, that's what your phone uses to classify your pictures into the pictures of your friends, for example. And in that case, what we would use is its capacity to summarize that global picture into a set of numbers that characterize, whether it's inside or outside or next to a certain structure. Okay, so by using these two things, we hope to extrapolate from a relatively small number of points the information to the more global ocean. And with that, I thank you, and I think it's time for questions.
Thank you very much, Stéphane, Pavla, and Jean-Olivier. Yes, we open now to some questions, so feel free to raise your hand or write anything in the chat. Maybe while you think about any questions, a useful piece of information is that the demonstrator will be live, accessible in a few weeks, so probably in June. So we invite you to sign up to the Blue Cloud newsletter to get notified when we have the official launch date of the demos. So so far all the information is available on the website, and of course in case of specific needs you can always contact Blue Cloud team. Is there any questions so far? Yes. So we have one question from Yvan Deprat. Is it possible to have a list of Blue Cloud resources with the URL mentioned there, the data catalog, the notebook, and so on, so it can be easier to test it? So I'm not sure if anyone wants to reply in the meantime. So I might say so that, so feel free to reply in. So this is the link. Let me paste it in the chat for everybody to to the page where you can access the lab, and from the lab, you will be able to access the lab. And from the lab, you get also the resources, but Pavla.
Yes, I think when the final, or when the demonstrator will be online, all of this is accessible, and it will be quite easily accessible. So there should be no problem in accessing all of the URLs, as well as the source of the data, which will be linked to the EMBL source. So even for all of the raw files, we will provide information where we got them from, as well as several publications where these data have already been published. I think if that answers the question.
Yeah, thank you, Paul, and thank you, Ivan. Any other question? No? Okay, I think in that case, that's probably it. So as I said at the beginning, this was one of the series of webinars that we organized about the Blue Cloud demonstrators. So in case, you've missed the previous ones, they are all here. And there is another question in the meantime. So how do you gather the information on the 150 stations?
I don't know. The 150 stations, well, we have gene catalogs for these, and we have environmental data for all of them, and they are partially already published, as Stéphane said, in Pangea or at the EMBL. So we're trying in the Blue Cloud to combine all of these data so we have them easy accessible for researchers that are focusing on specific research questions. And I mean, how did we gather them? It's published in many of the reviews of Tara, how they've been sampled, if that's the question. I'm not sure. Do you want to add anything, Stéphane?
That's pretty much it, depending on what is meant by gathering.
Yeah.
So if you want to get the raw data, then as we said, these are in open access at ENA, and you get the environmental metadata and contextual data at Pangea. But if the question was about when we use the Blue Cloud demonstrator, I guess the answer is that the subset is in the Blue Cloud, right? It can be directly downloaded and accessed through the Blue Cloud. Can you confirm?
Yeah. Yeah, thanks. So raw data, yes. And so there are some questions coming in. So Georgios Koutulas is asking, is this a training tool only, or analysis tools can be afforded eventually implemented on compute capacity?
Yes, so for now, this is a training tool. Or for now, it is a tool that is only used on a partial part of the data as a demonstrator. The idea that we are discussing already is to have a larger amount of more space in order to be able to run more analysis and or have access to larger data size. But for now, it can be a training tool only.
Yeah, but I think this is just a temporary issue until the demo is live, because all the areas are thought to be accessible and usable with the existing data or open to any further implementation in terms of other data and other services to be linked to that. So please consider this as just as a temporary thing. Another question from Ivan. So considering artificial intelligence, is it some tasks that can be of particular interest to propose for a challenge? I'm coordinating data access to biodiversity data sets for such a challenge in France and search something around Terra open data in the Mediterranean, for example. I think, is this referred to hackathons, I presume? Yeah,
I can probably answer that. So I know exactly the challenge that Ivan is mentioning, and the Tara community is looking into proposing something for that challenge, indeed. So one possibility is indeed how do we link local information, local really precise information that we get from a ship to more globally available information, such as what we get from satellites, for example. Okay, so that's typically a really good challenge for which artificial intelligence method may be deployed. For the particular challenge that we're talking about, which is something funded by the ANR and a gift from Facebook, I think there's some conditions about the fact that it should be about coastal sites, coastal meaning 200 miles from the coast in the Mediterranean and in the Pacific Ocean. So in that sense, Tara can contribute, but if you limit the stations, if you limit the Tara dataset to the stations that are in the Mediterranean and in the Pacific, within 200 miles from shore, it's actually going to be a quite small number, and artificial intelligence requires large numbers to do something. So it might be a piece of the puzzle, but it won't suffice by itself.
Thanks, Jean-Olivier. Maybe if you can just write down a couple of lines to the answer, that would help us in recording this question as well. And we have another one by Ivan. So next steps are also include the possibility to have more different time periods, variations, seasonality of models, for models. I'm not sure, Pavla, can you answer that? Or Jean-Olivier?
Well, yes, since it's referred to models, I'm assuming it's about the last part. So it comes again to the problem of the amount of data. Okay, ideally, what you would want is to link the observation from Tara that was done on the 25th of June in that particular location to the conditions on the 25th of June in that particular location, and extrapolate that to everywhere in the world and the rest of the year. And of course, you can do that when you have thousands and thousands of it's really difficult to do it from a smaller number of observations. The second aspect that is less of a technical problem, but it's the fact that the plankton that you sample in a particular location at a particular time has not been sitting there for its entire life. It's actually coming from elsewhere and living and moving towards somewhere else together with the water mass. So the plankton community that we collect in a certain location is actually representative of a broader ecological context rather than that precise location at precise time. So even if we had all the data in the world, it would still be worth not focusing on a really precise time and place, but enlarging the time window. So so the answer would be somewhere in between, basically. So we don't want to go to a really specific time period. Ideally, we'd like to be a bit more precise than just taking climatologists that average the whole year. So indeed, maybe we're going to try to go towards seasonality. So instead of associating that observation with the average temperature at this point over the whole year, we might link it with the average temperature of that particular season, because we know that at high latitudes, at least, the seasonal signal is really strong. So we might go to season, we won't go any further, like to months or something, because again, I don't think we have the data, and it might not even be relevant for the type of organisms that we're going to look at.
Thanks, Jean-Olivier. Any other questions? So I've also linked directly our LinkedIn page and the Twitter profile, since it would be nice if you could also help in getting us in connection with any other genomics researchers, for instance, interested in this kind of sourced services and tools. And so with that, I think that's it, unless there is any other burning question. Okay, so thank you very much. I remind you that the recording of the webinar, together with the questions, will be published online and accessible to everybody. And that's it. I mean, thank you all our speakers for the presentation and all the participants for your interest. Keep on following us, and we look forward to notifying you for the launch of the new services.
Thank you.
Thank you very much. Enjoy your weekend. Bye. Bye-bye.
Thank you. Goodbye.